This next presentation is about a company that was actually generated from an experience right here in Westchester. I'm going to let Greg Huntress introduce our speakers and talk about the background.
Thanks, everyone. So this is an exciting day for me for two reasons. Working with Michael and Joanne to bring us to the point where these kinds of scientific symposiums and discussions and business and entrepreneurial topics are being discussed with increasing regularity is exciting. The other reason for me is I get to spend 5 minutes introducing two really smart people who are going to give you a fabulous presentation, and then I sit down and I'm done for the day. So this was a bargain for me. The relationship that we're going to describe and the process of working together that the Jackson Laboratory, which is a nonprofit 501c3, with a biotech company to solve challenges around a rare and orphan disease, this can almost be viewed as a case study. And in particular for this audience, we had a basic science presentation. We've had discussions of how to interact with the FDA. We get to talk about the space in the middle called preclinical, which can be all the way back into in vitro systems. But at the Jackson Laboratory, we're focused around in vivo, in life, in animals, particularly mouse models. And there's a reoccurring theme that I've been seeing in the last 4 to 5 years as I work with more and more foundations and nonprofits that come up as patient advocacy groups, even things as simple as resource sharing groups. Oftentimes, parents of rare and orphan diseases start organically to communicate with each other. They organize that structure. In the case of chondrial, a family with the ability to invest in research becomes part of that discussion, and a company is formed. And the conversations that I have often are with someone who might be in what I would say more chemistry side. There's a target, we can show we hit it. Rick, we just need to run that quick mouse study, and then we're going to be at IND with the FDA. What are we talking, like $5 grand in 3 weeks? Can we do that? And what these folks learn, and we're hoping to convey to you today, is that there's a big space in this drug development gap between target validation and your IND. And there are a lot of mistakes that get made in this space. And I'm going to take 2 minutes to talk about the biggest one and the biggest change that we're seeing in this early drug efficacy testing. When you talk to a clinician, and actually I want to quote Jules because he did describe it very nicely. What's important to the patient is important. What's important to the clinician is important. What is the biggest challenge in the preclinical modeling space is oftentimes neither of those things are relevant to the science around the mouse model. A gene defect in a human, you put the same gene defect in a mouse, you don't see the same disease. So if your readouts from your study are the same clinical indications that the patient displays, you completely miss opportunities to demonstrate that your drug has an impact on the disease because you're measuring the wrong thing. I have a very simple example of this. We have 2 strains of mice called OB and DB. They have a defect in the leptin receptor signaling pathway. They get fat. They get fat like a snowball. They look like white baseballs. They get to the point where they can't move, and you have to put food on the cage for the bottom. For 20 years, biotech and pharmaceutical companies have been studying these mice for obesity, cholesterol metabolism, and cardiac disease. What percentage of overweight Americans have a leptin gene disorder? It's infinitesimal. I've been in the space for 20 years, and I have had one conversation with a pharmaceutical that was focused on leptin. I get asked this question probably twice a week. What is your best mouse model for autism? What is your best mouse model for Alzheimer's disease? These are spectrum disorders. The causes in the patients are wildly different. Kat and I had a conversation maybe a month ago with a group out of maybe 2 months ago. Time goes too fast. With a group that was very interested. This was a legit biotech company that was studying the ability of cannabinoid-derived drugs to impact seizures. Mice don't get seizures. You can induce a seizure using sound waves. So that's the study they proposed. And we're on the phone saying, what's the relevance of that to a severely autistic child who has 15 seizures a day? And the response was, well, we just need data to give to the FDA that shows it impacts seizures. So those are my examples. So the discussion that takes place between the Jackson Laboratory and companies like David's, they come to us, here's the gene, here's the clinical signs. We can make the mouse, that's just a tactical. But understanding how that gene works in the mouse model, and when you drug that, what are the readouts? If the mouse doesn't get sick, how do you know that your drug had an impact? If the mouse gets sick in a different way and your drug doesn't impact that, how do you know? By partnering with folks who have expertise specific to the technology that you're trying to implement into your drug discovery platform, you gain. I'm not a Ph.D. scientist. I do have a science background, so I'm gonna say this with all the love in the world, but really smart people have blind spots too. And that's what this conversation is about. It's about acknowledging there are blind spots and bringing expertise in. Folks, you can avoid the common traps. So with that, that's my 5 minutes, I think.
Okay.
Did I earn my pay for today?
You did. Good.
So I'd like to introduce our two speakers. Are you going first, Kat?
I am.
Okay. As you can see, I have some rapport with Dr. Lutz here. Just have to find you in here, Kat. Actually, this is, so Dr. Lutz has spent over 25 years in the field of mouse genetics and works with patient foundations to develop mouse models for the research community. The program at the Jackson Laboratory includes a growing collection of 10,000 genetically engineered strains of mice, with over 1,700 of these as live colonies available for drug discovery and drug research. It's a much longer description of Kat's.
I was going to say, I'm surprised you had to read that.
Yeah.
But that's fine. Thank you.
So with that, and actually, let me introduce David so we can do the transition quickly as well. So this is a two-part presentation, by the way, in case I didn't introduce that well. Jax is going to go first, and then Condra is going to talk after. So Dr. Bedouin is a drug discovery and development expert with extensive experience in preclinical research and development, global research strategy, and startup management. He has over 17 years of experience and a proven track record of achieving qualifiable results, implementing effective and highly successful internal and or partnered R&D programs to further drug development for both small molecules and biologics. He is passionate, and I can testify to that personally about what he does, about providing groundbreaking approaches and solutions for early and mid-stage programs. So with that, I will hand it over.
Thanks so much. Thank you, Rick, and to the organizers for inviting me to speak here today. I'm going to tell you a little bit about what we're doing at the Jackson Laboratory. I'm going to give you a little bit of my perspective on rare and orphan diseases, especially in the preclinical world, where we've been, where we are, where we're going, and hopefully engage us in a conversation around resources and how we get to where we need to get to and how we do that most efficiently and effectively. So as Rick told you, I'm a mouse geneticist, have been so for as long as I've been walking, and that's the only reference to my age that I'm going to give you. So you know, in the past we used to cut our teeth on positional cloning and gene identification, and the technologies have really brought us a long way over the years, and so I want to talk about how we can incorporate new technologies and get to where we need to be. I'll just give you a little bit of a flavor, again, for what we are. Jackson Laboratory is mouse. We're not rats, we're mouse. We were established in 1929, philanthropically, go figure, we're still around. And we live, the institution is on an island off the coast of Maine. And so I can honestly say I probably work in the most beautiful place in the world, right in Acadia National Park. We also have campuses in Sacramento and Connecticut as well, for some of you Connecticut friends who are here. It is a beautiful place. It's a little cold sometimes, but the one thing I like about Jax, and I'm not just showing you this picture because it's a beautiful picture, but the people who live and work in Bar Harbor do so because they really love the area. I've been there for over 25 years. And so the depth of institutional knowledge that is at Jax is just not comparable to many of the institutions. And so as a result, you know, the rich history and knowledge really makes us unique. I think we already talked a little bit, you know, we're known for our resources in mice, but we're also known for our faculty and our research. And it really is this union and intersection of being able to do everything that we do with mouse resources that's predicated on the research that we do at Jax. And I can honestly say that there isn't a single resource that we offer from reproductive biologies to CRISPR technologies that really just didn't stem organically from the research that we do. So that's also kind of interesting. We're not just adopters, but we're innovators as well. So if I can move to rare diseases for a minute. I think we all have a story and we all have a passion, and I think for most of us we probably have more than one. And it was really for me when patients started coming to the Jackson Laboratory and asking for our help in research. As we know, I'm not going to really read you the stats, but a lot of what has happened over the years is through exome sequencing, patients are being diagnosed more and more. And as a result, you know, maybe they took 5 years or 6 years or some incredibly long time to find a diagnosis, and then they find a diagnosis and they realize there's nobody working on their disease, there's no resources, and they come, you know, to us, you know, to try to find solutions. And honestly, we really do want to help. And I think, you know, it's a little sad to me personally that so many families have to self-assemble to fund their own research. And I really wish there was a way that we as a rare orphan disease community could streamline this process. I probably get a phone call every day or every week from a family who says, my son just got diagnosed, my daughter just got diagnosed. You know, can you help us? And this is an article that was in the Wall Street Journal recently with a family that we worked with. This is Luke Rosen and his daughter Savannah. And it's an interesting story because it came to us, somebody in my communications office called me and said, do you know this guy? I said, never met him before in my life. And he said, well, he's got a GoFundMe, you know, campaign going on because he said that his daughter was diagnosed with a KIF1A mutation and that the Jackson Laboratory was going to make a mouse and they needed to raise $40,000 to make this mouse. So long story short, we reached out and we got on the phone and he was just quoting numbers, you know, from someone, you know, that he got information from and said, yeah, it probably costs like $40,000 to make a mouse model. And then he told us a little bit about his daughter and what the disease was, and it turned out that we actually had an NIH precision genetics grant already in place that could fall really nicely underneath this. And I said, please just save your money, you know, we'll make the mouse for you, take the money that you have and use it for the research, you know, that's going to be subsequent that you're going to need. You know, and he was so grateful and so thankful, and I can tell you we have made the mouse. Not only have we made Savannah's mutation, but we've made a number of other alleles from these patients. And it just seems to me, and it strikes me, is that this is the way that it should be. You know, it shouldn't be patients raising their own funds. And I know as a group, we're assembling, NIH is doing a lot to fund rare and orphan diseases, but the rate at which we've diagnosed patients to the rate in which we gather these resources, there's a huge gap. And we're really interested in closing that gap. So the other thing about, mouse models, I think everybody understands when we work with patients and foundations that, mouse models are an important part of the process. We're not going to do, we'll do natural history studies on patients, but we're not going to be able to answer some of the pathology questions that we need to in patients at this point. Many times, the patients and the diseases are so new that we only have a handful. We don't have any post-mortem data. How are we really going to figure this out? And so mouse models are really important in trying to help us understand the pathophysiology in the mammalian system. But they're also very important for us preclinically as well, because when we do go to the FDA with an IND or a pre-IND, they usually want to see some sort of efficacy in a mouse model. That really helps the cause. And so getting these mouse models is really important. But if there's one thing that I've learned, for doing this over the years, and I've been doing this for a lot of years now, there are sometimes barriers to the effective use of mouse models. We all know that we need them, but how do we get them? And I think one of the things that we've come up with blockades on so often is accessibility and who owns your mouse. So you're a patient foundation and you fund researcher X at the university of wherever, and the person who owns that mouse is their tech transfer office. And often they'll put licensing around the mouse model. And so with all the good intentions of the foundation and the researchers, you really have to think about pre-negotiating the rights to those mouse models, when you're looking to collaborate with external folks. And we've come across this over and over again. A lot of the mouse models that we make today, we're just remaking models that exist because the person who made them will not give them up beyond their laboratory. And that really, I heard a groan. I'll groan with you because it's really sad. It doesn't help the patients. We understand that there are graduate students that have to graduate and postdocs that need to get funded, but this is the wrong mechanism for us to move fast and quickly in rare diseases. So I'll just put that out there. And so, we need to be efficient, we need to scale, we need to have standardized outcome measures for a lot of these. Reproducibility is a big key factor in this. How many times have we picked up a paper and someone said, here's the outcome measure, we get the mouse and we say, we don't see it. And there's a lot of reasons, and it's not bad science, but it can be environment and it can be a lot of things. And so, having those mouse models out there in the public domain and being used by a number of different groups who can validate them is really important. And so if I could just talk a second about the mission of the lab and what we're trying to do in the Rare and Orphan Disease Center. It's multiple, right? We want the mouse models at JAX. We want them in the public domain. We want anybody to be able to call and get a mouse model, and that's it. There's no MTAs, there's no licensing agreements. A lot of times we're still dealing with that, and we really try to discourage that process from happening. But at least they're accessible, and you don't have to, get them from a lab half across the world, and they're growing the mice in the sewer, and the health status is all wrong, and you can't really do anything with it. So that's always been our first mission, is to make sure that the mice are accessible to the biomedical community. And then for whatever mouse models that aren't there, we can engineer them. And I think the saddest thing about not having the mouse models that are already out there in the public domain is genetic engineering is not hard, guys. It's just not. It's not 10 years ago when it took 4 years to make a mouse and have it published. You can make a mouse in 10 weeks, flat, that's it. So people who are hanging on to their mouse models thinking they're liquid gold, they're not. And we really don't want to spend our time and resources remaking mouse models that already exist, but we will. So the other thing that we do is we partner with foundations and pharmaceutical and biotech companies, and I think this is really the crux of the meeting and the topic today. A lot of times it's very collaborative. We work from scientist to scientist to try to understand the mouse, the pathophysiology. But we've also set up a system quite nicely where we can provide services so that your intellectual property is your intellectual property. It's a fee for service. You ask us the question, and we provide the data back to you. And that's a lot what we've done with Chondrial, and I know that David's going to describe that a lot in detail. But again, this is where I think that we really provide the biggest service to the scientific community. You don't have to collaborate with me. I don't even have to know your name. All you have to do is go to the Jackson Laboratory, go to our catalog. You want a mouse for Huntress syndrome? Go get your mouse for Huntress syndrome. You want it for Friedreich's ataxia, SMA, whatever the rare disease is, these mouse models are accessible to the entire biomedical community. And I think it's something that is very unique and something I'm very proud to be a part of the Jackson Laboratory because of that. I'm going to tell you just a little bit of how I got started, and I got started in SMA. And when you work in mouse models and preclinical work and you work in rare diseases, I don't think, at least for me, I didn't think there was going to come a day where we were really going to have a drug that was going to help these kids who, by all purpose and pretenses, when you're a type 1 patient, you're going to die by the time you're 18 months of age. And so working on this was very powerful and critically needed, and it wasn't hard for patients and parents and foundations, to really convince us that this was something worth working towards. I just never thought we would do it as quickly as we did it. And I do want to say, for those of you who don't really know the story about SMA, I'll make it really quickly, but this is our story. The foundation approached me in 2010. We made mouse models, we characterized them, we did a lot of preclinical testing, we worked with a lot of drug companies, and we said, it's not working. And they said, are you sure? And we said, oh yeah, we're sure. Because we knew these mouse models so well. And the other thing is, we don't have a dog in the fight. If your drug isn't working, we're going to tell you it's not working. I have no intellectual property. The mouse isn't mine. I'd love to see the drug work, but I have no financial investment in that. And that's important. We're a third party, completely independent, honest broker, if you will, of the data. And so I think, as you all know, in 2016, the FDA approved Spinraza, and we have some really nice gene therapy with Avexis coming down the pipeline. And this is a real, success story. And let's face it, we swung for the fences on this one, and we hit the home run. But there's a lot more home runs out there. I'm convinced of it. We can do what we did in SMA for other rare diseases better, faster, and stronger. I'm convinced of it. So that kind of brings us to this pipeline overview that we're working with currently. We're not just a mouse model repository. If you want mouse models, like I said, call us up. Go to our catalog, get what you want, and be happy in your research, and we'll wish you well. But we can also work in so many different ways. We phenotype the mice. Natural history data on patients, there's natural history data in mice that we have to get as well. We have to identify, what are the most clinically relevant outcome measures that we're going to see in the mice that we can apply to the patients. And we also need to identify for our industry partners, what is the fastest indication? What's going to tell you, no go, we're done. It may not be the most clinically relevant, but it'll be the quickest that we can give you. So we work with our industry partners to try to get that right balance of, we don't want to waste a whole lot of time and money if we're going to fail fast. If we're going to fail fast. But we also want to make sure we're looking at things that are most clinically relevant. And so we have these phenotyping and testing paradigms and these outcome measures that we look at. And I put that little hospital in the corner because the Jackson Laboratory really is a mouse hospital. There isn't anything that we can't study, diagnose, you think about histologically, behaviorally, what we have for proteomics, MRIs, diagnostic histology, it's all there. And this is really important for biotech companies as well, who may not have those kinds of facilities in-house. We have it all. It's right there. And so if you're biotech, and whether you have a lab or you're virtual, it's going to be, I think, hard-pressed for a lot of folks to have the resources that we have at JAX. And so, again, we're pretty agnostic about it. You can use us as much as you want to or as little as you want to. The partnerships are not exclusive. We're just here to help. And so I'm going to talk a little bit and introduce, and then let David take over some of the things that we've been doing in Friedreich's ataxia. Because, SMA was interesting. I spent, probably 5 years trying to make a type 2 SMA model, something that really represented type 2 kids. I think we were successful in doing that. I'm not necessarily sure at the end of the day it was necessary. But Friedreich's ataxia is a different beast. For Friedreich's ataxia, we don't have a lot of mouse models that really recapitulate the phenotypes of the mice the way we really want them to. And I'm just going to say that Friedreich's ataxia is really another one where I think that we can swing for the fences and hit a home run, because there's so many different ways to cure this. So many different ways that you can attack mechanistically and therapeutically ways to treat this disease. And so David's going to tell you a lot more about this, but essentially it's a trinucleotide repeat disease. It's not a disease of no protein, it's a disease of low protein. And so you can think of a number of different ways in which you can just boost the level of protein by replacing the protein, by stabilizing it, gene therapy, RNA, transcriptional boosting, so many different ways to attack this. But the mouse models are a little bit hard, because while humans do not tolerate low frataxin levels, mice are quite happy with low frataxin levels. And so we've had to take some unusual measures in order to get a mouse model for Friedreich's ataxia that we can use effectively in these therapeutics. And I know that David's going to tell you about that, so I won't spend too much time on it. But only to say that this is Ron Bartek. He is, I don't know if any of you know Ron in the rare disease community. And for those of you who do, you're probably smiling, just as widely as I am. Ron came to my lab over a decade ago and said, I need your help. And I said, yeah, it's really hard for us to deal with patient, foundations. A lot of times they come to us, they don't have a good scientific advisory board. Managing expectations is hard. And he went, hang on a second. And of course, Ron came to the table with all of those things. And I think that he has really epitomized what a rare disease foundation can do, and how you can really get the communities to rally around you. He's an amazing individual, and if any one of you have ever spoken to him for more than 5 minutes, you would walk off the cliff if he asked you to. I mean, he's just, he's really a convincing and wonderful person, and I'm very privileged to have met with him and worked with him, for all of these years. So with that, I can tell you that we've been working really hard. I've been working really hard for Ron. I don't want to let him down. We've had a number of different mouse models that we've imported from collaborators all over the world, and some that we've made ourselves that David's going to tell you about. But what I really want to end with this slide, I know David's going to talk about Friedreich's ataxia, and I talked about SMA, and I talked about success stories, but I want to leave you with this one thought. Insert the rare disease model into that pipeline, because we can do it. We can make progress in all of these different disease areas. If we have the right mouse models, if we have the right space validity, if we have the therapeutic outcome measures, we can take this and make this not one disease at a time, but really make this a pipeline for rare diseases. And that's where I'd like for us to think about going as a community. So with that, I'm going to ask David. Where is he? David. Oh, there he is. He's hiding in the back. To come on up and tell us about his work in chondria.
One just quick question. Could you explain what making a mouse entails?
It entails CRISPR-Cas9 technology. And so when we genetically engineer mice, what we like to do is we like to take the precise genetic mutation that we find in patients, and we like to engineer that nucleotide or amino acid change or whatever deletion might be into the mice. And so there's technology recently, it's not new anymore, but it's really reduced to practice where you take CRISPR-Cas9 is an exonuclease, you cut the regions in the mouse like a pair of scissors and you insert what it is you want. It's not ES cells anymore, it's not homologous recombination, it doesn't take 3 years, it takes 10 weeks. And so making mouse models is really, really easy and efficient to do. So when a patient comes to us and says, can you make my son or my daughter's precise mutation? The answer is absolutely yeah. Not sure we need it sometimes, but in a lot of ways we do. Because a lot of times these patients have missense mutations or point mutations. They're not complete knockouts or loss of functions. And so we have a lot of mouse models that are loss of functions. They don't necessarily help us. And if the therapeutic is an antisense or a read-through or something along those lines, then engineering that precise patient mutation is really important. So, did that answer your question?